Let's look at what is DNA or deoxyribonucleic acid. DNA is a hereditary material. It controls the properties and potential activities of a cell. It is made up of four kinds of nucleotides, namely D-adenosine monophosphate, D-guanosine monophosphate, D-cytidine monophosphate, and D-thymidine monophosphate. These nucleotides are united with one another through phosphodiester linkages in a specific sequence to form long chains known as polynucleotide chains. Two nucleotides join together to form dinucleotide, whereas three join together to form trinucleotide. Nicotinamide adenine dinucleotide, abbreviated as NAD, is an example of dinucleotide and an important coenzyme in several oxidation reduction reactions in the cell. In 1951, Erwin Chargaff provided data about the ratios of different bases present in this molecule. This data suggested that adenine and thymine are equal in the ratio, and so are guanine and cytosine, as below in the table 2.2. Source of DNA: man 30.9, adenine 19.9, guanine, thymine is 29.4, cytosine is 19.8. Rest are also listed. Morris Wilkins and Rosalind Franklin used the technique of X-ray diffraction to determine the structure of DNA. At the same time, James D. Watson and Francis Crick built the scale model of DNA. All the data thus obtained strongly suggested that the DNA is made up of two polynucleotide chains or strands. The two strands are coiled around each other in the form of a double helix. Coiling of two strands is opposite and they are coiled in anti-parallel to each other. Two chains are held together by weak bonds, hydrogen bonds. Adenine is always opposite to thymine and guanine opposite to cytosine. There are two hydrogen bonds between A and T pair and three hydrogen bonds between G and C pair. The two strands are wound around each other so that there are 10 base pairs in each strand of about 34 angstrom units. The amount of DNA is fixed for a particular species as it depends upon the number of chromosomes. The amount of DNA in germ cells, sperm and ova is one half of that of the somatic cells. Red blood cells have 2.3 in chicken and crab, 3.3, liver cells 2.4, 3.3, kidney cells 2.4, 3.3, and sperm cells 1.3, 1.3. All the information for the structure and functioning of a cell is stored in a DNA. For example, in the chromosomes of bacterium E. coli, each of the paired strand of DNA contains about 5 million bases arranged in a particular linear order. The information in those bases is divided into units of several hundred bases each. Each unit is a gene, a unit of biological inheritance. The E. coli genome consists of 4,639,221 base pairs, which codes for at least 402,088 proteins. Haemophilus influenza is the first microbe to have the genome completely sequenced, and this was published on July 28, 1995.